The brain, the seat of our thoughts, our perception and our feelings. Observing its nerve cells in a state of highest activity, one can see nothing. Invisible to our eyes, nerve cells communicate with each other by means of electrical pulses. The electrical charge at the outer side of a nerve cell changes within a few thousandths of a second, becoming first more negative, then positive, before returning to its original equilibrium. At the University of Freiburg, Professor Ulrich Egert and his team want to answer the question how thousands of nerve cells use these electrical changes to communicate with each other and how they form networks. Being the building blocks of the nervous system, understanding individual nerve cells is an important step towards this goal. Patrick Dini is one of his PhD students. A physicist by training, his background helps him to answer this biological question.
You could record the electrical changing within a nerve cell by inserting very fine glass pipettes within it. But this method injures the cell. Moreover, you only measure at the place where you inserted your pipettes. This is a limitation, especially if you want to get a more detailed picture of the cell activity. You need other techniques to learn more about the activity footprint that a nerve cell leaves within a network.
To this purpose, tiny laboratories have been developed, containers in which nerve cells can live for several months and build connections. The bottom of such a container is equipped with hundreds of contacts. Even smallest changes in electrical potential at these contacts can be measured. In the group of Professor Hirlemann at the Swiss University ETH, these so-called microelectrode arrays have been further developed, their device packing more than 11,000 electrodes within an area of less than 4 square millimeters. However, this does not automatically let you observe the activity of one specific nerve cell. The scientists have to solve a tricky problem.
We have these 11,000 points where we can measure electricity, which sounds like an enormous number, but we are looking at thousands of nerve cells sitting on the container floor. So one contact will record the signals of several nerve cells. We somehow have to assign the signals to individual cells.
In Freiburg, scientists from very different disciplines cooperate to solve these kinds of problems. Maxime Ombard is a computer scientist and has developed a software that can carry out this kind of task.
Modern biological analysis produce vast amounts of data that cannot thoroughly be analyzed by humans in a reasonable timescale. Therefore, we need the combination of computer and bioscience for further approaches. At one contact of the electrode array we are using, the electrical activity of one specific nerve cell will always look similar, and at the same time it will look different from signals of other nerve cells.
This means that one particular nerve cell will show up in the recorded data with a signal curve that is recognizable and roughly stays the same. Maxime Ombard's software analyzes these voltage curves that were recorded all over the contacts on the container floor. The program then attributes the signals to the nerve cells that generated them, and finally it reconstructs the activity of the nerve cells that form a network within the recording container. From this, the course of activity around an individual nerve cell can be analyzed and visualized, something that was impossible until now. 200,000 pulses of an individual cell were recorded over thousands of electrodes and combined in order to create these images of a living, working nerve cell.
This is what we call the electric footprint of the cell. That way, we can learn more about its internal dynamics and its contribution to the network activity.
And a better understanding of the nerve cells as the building blocks of the brain can lead to a more precise interpretation of the brain recordings that are used in medical diagnostics.